Thank you very much for coming for the virtual field day. My name is Yeshi Wameshe, Extension rice pathologist, and I have been in rice pathology for the last 10 years, and it has been very interesting seeing different diseases and also management options. And here we are at the rice pathology experiment field. And before I go to my topic of the day, I would like to say few things about what we actually are doing in our program. And we cooperate with industries, and we do some seed dressing chemical evaluation, and also some evaluation of chemicals and biologicals for sheet flight. And we do germplasm evaluation for resistance, and also we do some other aspects like coverage study for fungicide application, and we have got for sheet flight and for false mud. And some other things that we do is again evaluation of germplasm for resistance for straight head, and that experiment is down there closer to the building, and maybe when it's everything matures, you are invited to come and look at the varieties over there, and you can see definitively differences in the varietal resistance level. And the other thing that we are doing is resistance screening for blast. But we're not doing blast screening here in the open field. So we're doing it in pine tree, and the place is surrounded by trees. And we make that choice because we don't want to spread the blast spores to the farmer's field. So you can see how careful we are when we are doing our experiment. And we also do varietal screening and germplasm screening in the lab, in the greenhouse for leaf blast. And lots of materials, preliminary, advanced, as well as varieties are screened in the greenhouse for leaf blast. And we produce different kinds of inoculum for artificial inoculation in our lab, and we are busy almost throughout the season. And having said that about our program, and our program also is cooperating with other programs. For example, Dr. Jared Harkey, he plants ARPT and CRT across like 15 locations in Arkansas. And the plant pathology or the rice pathology group goes to all these locations to take disease data so that we can deliver the current resistance level of the varieties that you are growing, and make use of those data because we put them in the blog and also in extension publications. You find them everywhere. So try to make use of that disease reaction for the commercial varieties. That is very useful. And besides, we also go to the breeders field and evaluate their materials, the preliminary as well as the advanced materials, including the URRN, which is a regional nursery for different diseases. So we get our disease information from different directions. Having said that, then our main topic today is an integrated approach for disease management. And when I say integrated approach, it means that it's a collective approach. And you may ask, what are we collecting? Actually, what are we integrating? And there are three management tools. Number one, it is use of resistant varieties. Okay, if available, you should always go for a resistant variety for a specific disease. Okay, when I say specific disease, specific to your field history. So matching your variety to your field is very important. For example, in a field that has got blasts, if you are planting a variety which is susceptible for blasts, then that is actually wrong. So you have to go for a resistant one. But I'm not saying that you don't have to plant a susceptible variety if you know the variety is high yielding, but one important aspect is if you know how to manage the disease. I can tell you one story. I have been going to one farm almost every year. It is rice after rice field, and I always find blast over there. And that farmer was actually planting the same variety. Then one day I met him and I said, why are you growing this variety? Because it is very susceptible to blast. And he gave me an unexpected answer. He said, because I know the variety, it is a good variety, and I know it is susceptible, but I know how to manage it. So that is a good answer. So if you know your variety and your field, and then you know what to expect and you know what to do to manage the disease. For example, blast at the earlier stage, when that farmer was having blast at the earlier stage, what he used to do was just release the water and then increase the water depth. And then when it comes to the fungicide application timing, then he applies the fungicide at the right timing, and he gets the anticipated yield from that field. So he was satisfied with what he was doing. So if you can't find the resistant variety, then what you have to do is you may choose to go with a susceptible variety, but you have to know what is important disease in your field. That is very important. And there is no such thing like, a variety which is resistant to all diseases, okay? It may be resistant to blast, but may be susceptible for sheath blight. It may be resistant to maybe kernel smut, but very susceptible for fall smut. So, but knowing how to manage your variety is very important. And when I say to manage, then we come to the second point or the second tool of disease management, and that is to use best cultural practices. And what do we mean by best cultural practice? It starts with, choosing a good quality seed, and then I would, if it is early planting, then I would recommend you to use seed dressing chemicals, and particularly fungicide combined with insecticides. And then early planted rice so far we have seen are resulting in good yield with a minimum disease effect. So early planting is one other practice. And then seeding rate, the optimum seeding rate. Don't go with the maximum seeding rate unless you know that that field has got a problem of stand establishment. And then also you have to check about the nutrition of the field. And so there are so many aspects of best cultural practices that you have to do with when it comes to the disease management. And one big aspect of cultural practice is nitrogen rate. Okay, if the nitrogen rate is excessive, then the plant look very nice when it comes to the color, but they will be lush to be very susceptible for the diseases. So know the disease in your field and then manage it accordingly with the best cultural practices. And the third one is use of chemicals or biocontrol. And when it comes to that fungicide, and I have to emphasize that if we use fungicides when it's not required, then we are actually encouraging resistance, meaning that the pathogen becomes, in time, it becomes insensitive for the fungicide. And what would happen later on is, if we are thinking of the future, then we'll be out of any chemicals for the future use. So we have to be careful. And this is not like a new thing. This fungicide resistance or insensitivity by the pathogen is not a new thing. And pathogens are smart, and we try to be smarter than them, but they actually make their way somehow for existence. So they change their genetics to continue to live. And that's why it's called, that insensitivity or fungicide resistance is occurring. So we have to integrate these three tools: resistance, use of resistant variety, and application of the best cultural practices, and if needed only, the use of fungicides. And all the diseases in the field do not require fungicide. We have to weigh out which disease is the one which is actually causing the maximum yield loss, and which one is really important in your economics. So if, for example, it is blasts, you have to concentrate to actually manage blasts. And if it's sheath blight, and then you have to deal with that. Otherwise, we're not talking about zero tolerance in disease in your field. There might be some minor diseases that may not, if you actually apply fungicides, the cost of the fungicide would be higher than the loss by the pathogen. So because of that, so what you have to do is you have to weigh out, is it important? How extensive is that disease in your field? And is it economically important? So that, you can apply fungicide, and then, it may or may not pay the bill for you. So that is actually your judgment call, whether you have to apply for minor fungicide for minor disease or not. But the 4 diseases that require fungicides in rice are blast, sheath blight, kernel smut, and fall smut. And again, to minimize or reduce the cost, sometimes what you have to do is you have to pair the fungicides and the diseases, which means, for example, the kernel smut and fall smut protection is applied at mid-boot. Then, if you can delay your fungicide application for sheath blight until mid-boot and use a combination fungicide instead to target all the 3 diseases, then you are actually reducing the cost, at least the application cost. And again, if you delay further your sheath blight application with blast, maybe it is the same fungicide that may be serving for blast as well as for sheath blight, and then you'll kill 2 birds in 1 stone. So do not be in hurry to just apply fungicide so that you will just have a good night's sleep, and then but the cost will be high. So with that as introduction of integrated approach, then we have some experiment over here. For example, here on my right side, we've got about 340 entries. They are germplasm, URRN, ARPT, and some EMI lines, and some advanced breeding lines. And they are replicated, they are in hill plots. Hill plots means they are, here and there in 18 inches distance. And then we inoculate them artificially with the pathogen of the sheath blight, and we evaluate them later on. And one trick about this disease, sheath blight, when it comes to the germplasm screening, is we have to be able to identify and isolate that actually gives us the small difference among the varieties. If we take aggressive kind of isolate and then inoculate, and you'll see no difference at all. So everything becomes susceptible. But what we have to do is, usually we collect samples, and then in the lab we identify which one is aggressive, which one is a slow grower. So we actually take the slow grower one, and then we do the screening with that. And we can pick small differences among the germplasm. So when it comes to the sheath blight, there is no complete resistance. There is no like black and white. This is disease and this is no disease. There is always disease, but the tolerance level or resistance level is different. So there has to come, science and art together when we do this kind of thing. And behind me, I have got a ForceMat experiment. And this experiment actually was started like 3 years back. And it started with the sheath blight coverage study, which is 3 base below me on my right. And I've been talking about that last year. If you remember, I've been showing these bottles last year. And so we used 2 kinds of fungicides. They are premixes, Amistar Top and Quilt XL. And so we were comparing which one is good for sheath blight and also for ForceMat. And we also chose GPA, that is gallon per acre water carrier, to actually deliver the fungicide. And you can see over here, when we use the 10 gallon GPA, and even if the chemical is diluted, but the coverage is a lot. So a lot has been sprayed out. This is a 2-liter mix, okay? And then when it comes to the 3 GPA, then you can see how much it has been used. And so when we saw in sheath blight, definitively, in yield as well as in disease severity, there was no difference between unsprayed check and the 3 GPA. So if you are just using small amount of water volume to carry the fungicide during your fungicide application, then you are actually losing money, okay? That is in regards to the sheath blight, because we did the experiment and we have seen it, and it's consistent all 3 years. I've taken data already in that in those plots, and they have shown that a lot of similarity between the unsprayed check and the 3 GPA. But the 10 GPA, it is clean. So you don't see any lesion in those plots. So that makes some sense. And sometimes when I get a call at a farmer's field, and we ask questions, okay? So did you do this at the right timing? And what chemical did you use? And what variety is it? Is it susceptible? And what is the history of the field? And this kind of questions. And I personally forget to ask what water volume did you use. So, but after I ask those questions, and when I see a lot of disease, and when they tell me that they have done everything correct, then I'm like, what happened then? There could be some fungicide resistance. So I try to collect samples. But at last it comes to my mind, oh, okay, what volume of water did you use? And then they will tell me maybe 2 or 3 gallons per acre. And that answers the question. So if with that amount of water, it's not working, okay? Yeah, it's different. We actually used a Mudmaster for this one, but on a plain, at least the 5 gallon per acre to up to 7 gallon per acre would be reasonable. But if you are using the ground rig, then the 10 gallon per acre might be a good thing for you to go, okay? So based on our experience with sheath blight. But when it comes to the ForceMat, the story is different. So 3 years back, the first year, we didn't get any disease. So working on SMATs is very difficult, okay? So there was no disease. We actually collected galls, and then we crushed the galls and released with the flood water so that we can assume that we have artificially inoculated. And so it has got a history or so, and then there was no disease. So ForceMat is dependent on the variety, on the weather, and also on other conditions like particularly rain. So, but last year, what happened was there was a continuous rain late in the season. Maybe you remember the hurricane, the repeated hurricane rain, and for a length of time. And so the false smut was bad. By the way, the variety is diamond, so it is very susceptible for false smut. So it got bad, and I got excited. And then when the time came to take the data, then the first thing that we did was we put a quadrat and we harvested some, and then we started counting the number of panicles that has got or that have got the false smut. And then that result showed us the unsprayed check to be better than the sprayed check, whether it is 3 or 10. And I was like, what? So the fungicide is not working for this disease? And then we took again a second round sample, and then we counted even the kernels, individual kernels in a panicle that have been affected. And again, the result was the same. Again, the check was better than the fungicide. And then I was like, how am I going to report this? This is not as expected. So then I actually just walked in the field by myself alone to see what happened and to think what happened and what is the possible explanation. And I remembered one experience I had from tilt back home in Ethiopia. So I was working at that time on wheat, and tilt had a greening effect, meaning that it keeps the plants greener, for a longer period, at least a little bit longer. So I said, maybe that could be the effect. So I tried to look the difference between the plots, and I saw that greening effect on the sprayed plots. And then I actually started to think about the biology of the fungus. So secondary infection is possible. So as long as there are green tissues that in the kernel, then infection can occur anytime. So it's not only the primary, so the secondary. So if that is so, the question will be, should I use fungicide for controlling or managing false smut? And my results are not conclusive, so I cannot tell you, no, you don't. But I can tell you that the integrated approach, if you can find a better resistant variety for false smut, that is the way to go, okay? And then early planting, cultural practice, and again, adequate nitrogen fertilization would be a good way to go. So with that, you can with this integration, you can minimize the disease. So I can tell you that it's not conclusive, but that is my first year observation, and I don't know what would happen this year. So it's late planted, relatively normal late, towards the mid-May, and we'll see how it turns out. So, but the result was not conclusive. So you can actually start observing with me how it is doing out in the field. Is there that greening effect actually that you see, and do you see that more false smut on the secondary tillers than the primary tillers? Probably, what would happen is the fungicide might have worked on the primary tillers, but from the primary tillers, a few spores may drop on the secondary tillers, and because they are coming late and they are green, so they may get the disease. So that might be the case. So I want you to do that observation with me, and because working on smuts is not an easy work, and I wish I could give you a right answer for this one, but you will be the one who will be researching with me through the years, when it comes to the false smut. But for any disease, that integrated approach is very important. I'm a big believer of using the cultural practice, the best cultural practice, because in so many fields that Jared Harkey is planting with CRT and ARPT, I don't find that immense amount of disease, because he follows the recommended package for those varieties, and because of that, and disease may not be severe enough. So definitively, the take-home message is When it comes to the disease management, number one, it starts way before the season, starting with your varietal selection, and then the quality of the seed, and land preparation, and then knowing the history of your field, and then integrating or compiling all these approaches to combat the disease is very important. I'd like to say thank you to University of Arkansas System Division of Agriculture for the opportunity given to us, and I would also like to appreciate the funding from the Rice Checkoff and also the financial support from our industry collaborators. Thank you.